Okay, hi everyone. This is Chelsea. I teach a few of the sections for Bio 2 Lab. I'm going to go through the parts today. The newest version of Mega, the newest update is somewhat glitchy on a Mac at least. And hopefully if you have a PC and you're also struggling, maybe this will solve a few of the issues that you're having. I'm not quite sure that that will work or not. But either open up your manual to page 133 with all the instructions on it, or you can open up, let me just go ahead and share. You can also open Mega, so that PowerPoint online has all of the instructions. So you'll open your alignment, you'll open your file after you've downloaded it. So if you are on Canvas and you still haven't even started, you will have to download this FAS file. That's the actual sequence file. You need to download that first before you do anything else. You'll have to download Mega here, so the software on a Mac or a PC, whichever you're running, and it should recognize which one you need to download on your computer. And then you can open the presentation slides here or open up your manual. You will want to have those constantly up. This only takes a few minutes once you actually get rolling and know what you're doing. So it's pretty fast, and we'll do this again next week. So if you are having issues, the first thing would be to try going through this tutorial. If that's still not working, email your instructor with a screenshot of where you're getting stuck. Maybe they can help you walk through it. It's kind of last minute today, since the assignment's due today, but next week we'll be doing another thing with Mega, or at least one more time, maybe two more times during the semester. So, plan ahead, because you might want to use the computers in the lab. Even though we won't be in person for lab, we can still, if you contact us ahead of time, maybe get you access to that room. You can also try using a friend or family member's computer with a PC, but I'm just going to go through it today. I did get it working on my Mac, and I know it was a lot slower on my older Mac. So, maybe just give it 30 seconds with each step. It really should only take you 5 minutes total if you're going through the steps. So taking a little bit of extra time on a slower computer, it shouldn't take you more than 15 minutes still. So I had already opened this. Let me just go through this all again. So I have Mega on my computer. So it should look like this. You open the software before you open the file. If you double click the file and try to open it, it doesn't open for some reason. So you just need to open the program first, and you'll want to do edit and build alignment, and then you want to retrieve sequence from file. So you need to have it saved to your computer first. If you can see behind me, I already have it saved to my desktop. That's just the easiest place for me to find it. So it should be that FAS file. You hit open, and then the first thing that it's telling us to do from there is to select all. So really, if you look at it, only this first nucleotide is selected at this moment. So we need to select all of the nucleotides in order for it to know what to sort. So in this window right here, make sure that you have this window selected, and you can either go up to data and edit and select all, or you can just hit that shortcut, which is Command A. I just hit Command A, nothing happened. So this is the first weird glitch. As soon as I click on this other window back here, it is going to magically highlight all of my data on that other one. So I hit Command A, then you just have to click on this other window. It's doing it, it's just for some reason not showing it being done until you click on that other window. I don't know, it's a little odd. Go back to this window, come up here to alignment, and then align by clust all W. So that's one thing that you won't find on a Mac in this tab. You just need to go up to the top and hit alignment. So sometimes it's just a matter of trying to figure out what box to click. And we want align by clust all W, which is just the first one. And every step in here you'll see in your manual, it just says keep the default settings. So I'm going to hit alignment. Second weird part on a Mac, there are buttons down here. They're just for some reason not showing up, but they do exist. So I've just been clicking through these, and then I kind of click down here. The OK button's down there. It's just on the right-hand side. So if you click anywhere down there, that bar should show up. And so you can now see that this alignment has worked. And if I scroll through, I can see that those sequences are aligned as best as they can be. We have a lot of mutations, point mutations. And then at the beginning and end of these sequences, we have a lot of pretty large insertions or deletions, but everything is aligned at this point. So now I need to save my alignment. So on the bottom of page 134, I need to hit save. So go to the data menu and then save session. So data, save session. You can also hit Command S, just the shortcut. So we've aligned everything. We're going to save that again to the desktop. You'll see it's a new file extension. So instead of .fas, it's now .mas. I'm going to save it. And from there, you can go back to this main window. The last two things will be done in this main window, actually. So you can go to distance and then compute pairwise distances. This is the third weird part on a Mac. So it's this file right here with the .mas is what we want to use, because that's the one that we just saved, that we've already aligned everything, and we want to build our alignment from there. You need to click on options down here, and then it shows that all files is selected, but it's really not. So you just need to click on all files, and then hopefully, even though it looks like it's grayed out, if you click on .mas, it should work. If it doesn't work the first time, just hit cancel on this screen and try it again. So again, it's not letting me do it the first time. Hit options, all files, and then you should be able to select that .mas file, which is the one that it needs to be able to use. Just hit yes on this pop-up window, and then we're going to be using all of the defaults. So you can always just hit OK at the bottom. And then we have our pairwise distances. So kind of like what you made on your tables when you were doing the morphological stuff. Mega has just done all of that for you. The last thing that we need to do is create our tree. So we want to construct a maximum likelihood tree, which on your morphological trees, you want to have every trait on there one time. You don't want to have to put cuticle on your tree in 3 separate places. So Mega is now going to do the same thing when we tell it to construct a maximum likelihood tree. So that idea of parsimony, but we just want to select this top option here. Hit yes, you want to use the currently active data file. It should again be that .mas file. Hit yes. Again, we're keeping everything the same. This is that box that didn't show up on our first window, but it's working now. Hit OK. It should go really quickly. And this, pause the video here if you've gotten to this point and you want to check it with your own, because this is what you should get. It looks, I think, slightly different on a PC, and that's okay. We have all of the trees saved. We know all of the options of what you guys might get on your computer. As long as you get one of them, then you'll get the points for the assignment. Don't forget, on the bottom of page 136, it tells you to root the tree. I'm not going to do that here, but all you would do is you would choose which one of these needs to be the out group, and you would right click on the line going to that species, and then you would hit root. So you need to right click on it and then place the root on whichever one of those you think, and that then will be your final tree. I always just think that it's easiest to, on a Mac, hit Command Shift 3 or 4 to take a screenshot. So if I hit Command Shift 4, it allows me to just take a screenshot of whatever my final tree looks like after I've placed the root. So sorry, I realized afterwards that it does allow you to save the tree. I forgot about that. So if you go up here to image, you can save as a PNG file. And if you just click on the folder here, I'm just going to save it as tree. It just needs to know where to save it. So it won't allow you to save it if you don't give it a location and a name first. So just click on that folder, click OK, and then you should be able to open it from there. So I just saved it as a PNG because that's a really easy file type to just copy and paste into a Word document. Hopefully that covers all the glitches that you've been having on your computer. If it's still not working for you, email your instructor. and hopefully we can get you through it. But if it's just, one of these final steps that just isn't working, if you send us a screenshot of as far as you got, then maybe we can just send you the final tree. And then all I would ask is for you to be able to tell me where the root should be. Because that's kind of the final step that you should be able to do on your own. So hopefully that helped. If not, talk to your instructor. We'll do the best that we can. And in future weeks, plan ahead so you can kind of get into the lab and use our computers in there if your own computer isn't working with Mega. But other than that, enjoy the nice weather coming up and have a great rest of your week.